Okay, let's get going. Welcome. I'm Peter Atkinson. I'm the Dean of the College of Natural and Agricultural Sciences here at UC Riverside, and I welcome you to the Student Success Centre. If you haven't been here before, it's quite a large building, and the room that we're in is a real gem, and everybody should have the privilege and the opportunity to present a lecture or a seminar or even a town hall meeting from a stage like this, because you can actually walk around and see everybody in the class. And we have another one of these that is hemispherical above us, and we have a large number of other instructional rooms that can have portable tables, and that enables the students and the instructor, the professor, to rearrange the class as she or he sees fit. If you have time today on campus, if you go up to the top of campus at the sort of the summit of the campus drive, you'll see our new School of Business building, which is another delightful building to be in and to give seminars in and to host speakers at. And if you go not too far from us, I think in that direction, you'll see a construction site, which is our new fourth floor university teaching laboratory facility, which will be open in about a year's time. And if any of you have been in our Spieth Hall and biology laboratories, especially the instructional laboratories, and realise that they're the same age as me, but in probably a little better condition than me, but that's where we've been teaching freshman biology students for 67 years, and in a year's time we'll have these outstanding new laboratories to teach these students, along with a dance studio for our colleagues in CHAS, as well as organic chemistry laboratories for freshman OChem, and other instructional rooms for that are not laboratories. So the campus is moving ahead in leaps and bounds in the types of facilities like this that we can have these meetings in. So a special welcome to everybody, especially those of you who are here for Transfer Day. It's been a long day, and thanks for attending, and we've saved the best for last, if you will, which will be an event in our 4 series, Science Lecture Series. So this is the 4th and final for this academic year. The first 2 were in our Palm Desert campus in Palm Desert, and the 3rd and 4th have been here at UC Riverside. And today's speaker is Professor Adler Dillman, who is a professor of parasitology and nematology in our nematology department at UC Riverside, in which he is chair. Professor Dillman obtained his BS at Brigham Young University in Utah, then came to California to do his PhD in genetics at the California Institute of Technology, and then undertook postdoctoral research at Stanford University in Palo Alto. And in keeping with the Californian theme, we were very fortunate to hire him as a professor of parasitology and nematology in the nematology department. He's interested in nematode parasites, and I've listened to Adler before talk about nematodes, something that is not immediately obvious to most of us, but they are massively abundant life forms on the planet, and they are absolutely essential in many, many important ways, but they can also, on the negative side, be parasites as well, leading to sometimes quite severe diseases. He was awarded a very high honour by the National Institutes of Health, which is an award for an outstanding investigator. He is sponsored as well in his own laboratory by the National Institutes of Health, but it has to be an extraordinary investigator to be given the award of an outstanding investigator that he was given. He's also received the Chancellor's Award for Excellence in Undergraduate Research and Creative Achievement from UC Riverside. His lab, as I said, is currently funded by NIH. It's also funded by the National Science Foundation and the United States Department of Agriculture. And he recently reported the presence of a flatworm infection in dogs that and his field site was in the Colorado River, and that led to quite a lot of veterinary interest through Southern California and beyond in him identifying this particular parasite. He's been studying parasitic nematodes since 2004. We're privileged to have him as a professor in CNAS at UCR, and we're especially privileged that he accepted the invitation to speak at this, our fourth and final Science Lecture Series for 2025. So, Adler, please come to the podium.
Thank you. Thank you very much, Peter. It's a very generous introduction. It's a pleasure to be here with you today, to be here for the new transfer students, to be here with some of my colleagues and members of the Riverside community and the UCR community. I told Peter I love to talk about nematodes, and any opportunity is an opportunity that I would take advantage of that. So I want to just take a couple minutes to mention a few things about myself. So Peter gave a wonderful introduction, but I wanted to mention that as an undergraduate at BYU, I actually had two very meaningful interactions with UCR, and specifically with the Department of Nematology at UCR. So in early 2005, I saw an advertisement. I was an undergraduate at the time, and I had just gotten into a nematode lab the year before. So I was a junior, and they were advertising to get some students to go with a nematology group from UCR to the Republic of Moldova, where they wanted us to do some field work and study nematodes, but they also wanted us to build relations with the government there. And so I went in 2005 with Ed Platzer, who is now a retired professor emeritus in the Department of Nematology. But that was my very first experience with UCR and with the nematology department and with the wonderful work that they were doing. Later that same year, the undergraduate research advisor I had at BYU, Byron Adams, had invited a number of researchers to come sampling with him to a place called Black Dragon Canyon and Goblin Valley in eastern Utah. And among those he invited Jim Baldwin, who was a professor in the Department of Nematology. He's now retired. And his graduate student, Eric Ragsdale, who is a professor at the University of Indiana in Bloomington. And he still is a nematologist. So I had these two very meaningful experiences that kind of planted a seed in me about what a wonderful place UCR is and the exciting research that they were doing studying nematodes. By the way, I sometimes will switch back and forth between nematodes and nematodes. I hear it both ways all the time, and so I kind of like to use both of those terms. Peter mentioned that I went to Caltech, and I studied there. I learned about nematode genetics and genomics. What do you do with an entire nematode genome? How do you make sense of all that data? And when I went north to Stanford, I actually was studying fruit flies. So I spent some time studying fruit fly immunology and how insects deal with parasitic infection. And to my absolute delight, in 2015, I was privileged to start my lab here at UC Riverside. This is a picture of the first students that joined my research lab, and I could take time and tell you about each one of them and what they've done. But what I want to say is just what a treat it is to be in the Department of Nematology that I got to learn about as an undergraduate and get to continue that tradition of excellence in research and in undergraduate education. I also just want to take 1 minute to mention something about getting a college education. We live in a time where there's a lot of discussion about the value of education, and maybe in particular the value of a college education. And I just wanted to say that in my personal experience, my college education was extremely valuable, not just to my ability to earn a living, to make money. A lot of we hear talk about return on investment, but it has affected every facet of my life. Aside from being able to work, obviously as a college professor, I needed to get an undergraduate degree and a PhD. But I think that being in college has intrinsic value, and having that education has intrinsic value. And I just wanted to share a couple things that in my experience enhanced my education for me. And one of those is that I would encourage you to be on campus and to be involved on campus. There are so many activities that are planned for you, with you in mind as students. You should be here. We live in a time when it's easy to telecommute and to Zoom. I would encourage you, where possible, to be on campus and to be engaged in the activities and the social networking and the academics that's happening here. I would also encourage you to get into a study group. I know here at UCR we have learning communities. When I started college right out of high school, I had no idea how to study, how to take notes in class. I didn't realize that you actually spent more time out of class preparing for class and doing the research, reading and studying. Joining a study group changed the way that I interacted with my academics. I learned how to take notes in class, how to prepare, how to take exams. I would encourage you to do that. And then I would encourage you to seek out opportunities in your area of interest. My area of interest was science, and I mentioned 2 opportunities I had as an undergraduate. I was able to go to the Republic of Moldova with the group from UCR. That's an opportunity I got because I was getting a college education. I was able to go to national and international science conferences and present my research, meet collaborators and colleagues. Being in college allowed me to take advantage of those opportunities. You will have similar opportunities here, and I encourage you to look for them in your area of interest. I would encourage you to get to know the faculty, get to know a professor, or even better, multiple professors. Whatever your next step is for your career path, you're likely to need several letters of recommendation. Too often, students start thinking about those letters in their final year as a senior. That's not the right time to develop those professional relationships. You should be doing that when you come. You should take advantage of office hours. You should take advantage of getting to know the professors that are exciting to you and that you think would be interesting to talk to. Most of my colleagues are very friendly, and they're here because they're interested in undergraduate education and they want to help you advance in your career. So take advantage of those opportunities and get to know us. And lastly, I would just want to reiterate this point that I think your education has intrinsic value. Yes, it may allow you to earn a living and there may be a monetary return on investment there. But as I look at some of the classes I took, although I was focused in microbiology, I took art history courses, I took theater courses as an undergraduate, I took literature, and all of those courses have enhanced my enjoyment of daily life. Of course, I love to take walks and hikes, and my understanding of biology enhances my appreciation of the natural world, and it makes my life more enjoyable. I love going to the zoo, not only to see the animals there, but I have an understanding of some of the evolution that's taken place, some of the adaptations. My life has been enhanced. I can go to the opera, I read books, and my education built a foundation for me that just enhances my appreciation of life in general. So I just want to end by saying that I think your college education has intrinsic value. Okay, so now to move on and talk about nematodes. I want to take a few minutes and introduce you to what nematodes are. And after I've done that, I want to spend some time talking about a very particular group of nematodes called entomopathogenic nematodes. That's a subset of insect parasitic nematodes, and I'll introduce you to them. And then I want to spend the last few minutes of my talk sharing with you some of the research we're doing to address 2 questions that have been fundamental to my research program. One of those is, how do parasitic nematodes cause disease? And the other is, how do parasitic nematodes manipulate host biology? And we know a little bit about both of those things, but there is so much that remains to be discovered. And if your interest is in science, you could get involved. Not just in my lab, but in any of the labs that are here on campus. And I would encourage you to explore and look for opportunities to do that. So if you think back to a course that you've taken in high school or in freshman biology, we talk about categorizing life. And I learned a mnemonic. I learned kings play chess on fine-grained sand, right? Kingdom, phylum, class, order, family, genus, species. So, and of course, now we have this additional classification of domain that's even larger than kingdom. I don't want to get bogged down in those details, but I want you to think about the animal kingdom. So if you think about the animal kingdom, think about the members of the animal kingdom that you could name off. I would imagine for most of you, many of those that you would name all fall into one phylum, and phylum is the second largest classification, right? So you might think of things like monkeys or dogs, cats, bears, tigers, lions, and it's true, those are all animals. Nematodes are also animals, insects, spiders, slugs, snails. There are many animals that fit within the animal kingdom. And in fact, depending on the classification scheme that you use, there are 39 phyla in the animal kingdom. Most of the phyla, or most of the organisms you would think about, fall within one particular phylum, and that's the phylum Chordata. That includes amphibians, reptiles, birds, fish, and mammals. But look at all these other phyla that are listed there. Some of you may be familiar with arthropods, right? The phylum Arthropoda, that includes insects, spiders. You may even have heard of something like a tardigrade. That's a whole phylum of organisms. The phylum Tardigrada, they're interesting and charismatic. I'm going to talk today about the phylum Nematoda, and that is a phylum unto itself. There are about 30,000 described species in that phylum, and they're all unsegmented roundworms. Not related to earthworms. Those are in the phylum Annelida, and they're not related to flatworms. That's, again, in a whole nother phylum. So what are nematodes? Well, for the most part, they are microscopic roundworms. Now, there are some that are not microscopic. If you came to my office on campus, I have on display a couple of dissected examples of a nematode called Ascaris lumbricoides. And this is a nematode that is 8 to 10 inches in length and about the width of a green bean. And it lives today in about 1.2 billion human beings' intestine. So it infects a huge number of people on planet Earth, and they're hanging out in the small intestine. So not all of the nematodes are microscopic, but most of them are. And here I have a couple of examples. I have a parasitic nematode on the left, and on the right I have C. elegans, or Cenorhabditis elegans. This is the model nematode. We consider C. elegans a model system because it's a model for biology in general. We study ecology, neurobiology, development, genetics, genomics. We study every aspect of biology in this nematode worm, and what we learn about it is applicable largely to other organisms on Earth. As you look at the picture, I have a picture here from under a microscope and a diagram, and you can see it's naturally transparent. That top picture comes from under a microscope. You can see the mouth, you can see a buccal cavity, the esophagus, you can see the reproductive tract, you can see individual neurons in that nematode, and it has become a very powerful model system for a number of reasons, one of which you can house millions of them in the space of a drawer. Can't do that with mice. It is naturally transparent, as I mentioned. It has a very fast generation time. So they go from an egg to a fully reproductive adult in less than 3 days. So very fast turnover. You can generate large quantities of these. And they're only about 1,000 cells in terms of their size. So they're 1 millimeter in length, thereabouts. They're about as thin as a human hair, actually thinner. And because they're only 1,000 cells, their central nervous system is a subset of that, which means that their brain and central nervous system, it ends up being about 350 cells. So if we want to start to understand how brain cells are involved with regulating behavior, we can do that at this scale. There's only 350 cells in the central nervous system. So it's a very powerful model system. There's also been 4 Nobel Prizes awarded for research done in C. elegans and discovery of mechanisms and biological aspects that are applicable broadly to all or most of the organisms on planet Earth. C. elegans also is beautiful. So this, if you were to look under a microscope at C. elegans, this is what it looks like. It moves with this sinusoidal wave-like motion. This would also make a nice screensaver. And I think the reason that it was named C. elegans is because it's so beautiful. It's so elegant in the way that it moves. Now, again, this is under a microscope. It's about 1 millimeter in length. They're very small. C. elegans is one of about 30,000 described species of nematodes. And here are some pictures of other nematodes. They don't all look the same. They have interesting appendages and morphology on their mouth. Some of them have these textures and ridges that are involved in their particular lifestyle. They have evolved to occupy virtually every ecological niche on planet Earth. Here's an example of some that were discovered living in a cold methane seep on the bottom of the Pacific Ocean. I have 3 different specimens there. You can see one of them has a great big wide open mouth. One of them, it's hard to make out the mouth structure at all, and it's got all these elegant, beautiful hairs coming off of it. 2 of them, you can see a spiral-shaped organelle there, a sensory organ. That's called the amphid. It's like the nostrils on the nematode. It allows it to be able to sense the environment. But these are beautiful. This is done under scanning electron microscopy, and these were species that were discovered in 2014. It's believed, although there are about 30,000 described species, there's at least 100,000 and maybe more waiting to be discovered and described. So of all the species that have been described, only about 25% of them are parasites. That means that 75% are non-parasitic. They live in marine environments, in the oceans. Some of them live in freshwater environments. Nematodes like C. elegans live in the soil. They're involved in nutrient cycling and soil health. But of course, as scientists, a lot of our focus is on human health and well-being. And so we spend a lot of our time focused on those organisms that are parasites. The ones that are plant parasitic, some of them cause devastating crop loss in agricultural systems. And so a lot of research is done looking at how to prevent those infections, how to kill those nematodes, or how to control them. There are many that cause debilitating diseases in human beings and cause significant human suffering. An example I have here is Ankylostomia duodenale, which is a hookworm. This particular type of nematode, hookworms, infect over 700 million people on planet Earth today, and they can cause anemia and other significant pathologies that lead to human suffering. So, of course, we're very interested in understanding how these nematodes cause disease and what they do. As I mentioned, nematodes have evolved to occupy virtually every ecological niche imaginable. I have pictures here of just places that I personally have been to collect nematodes from the soil. Here you have in eastern Utah, Goblin Valley in the top center. I have 2 photographs of research studies that I've participated in Antarctica, looking for nematodes in the dry valleys where they're essentially the top of the food chain. There's no insects that live there or vertebrates, except maybe a wandering seal that makes its way in there and dies because there's nothing to eat. They're here in Southern California, Central Valley, Northern California, and in the Republic of Moldova. They live everywhere. And that's exciting because if you study nematodes, it means you could travel anywhere in the world and still be studying nematodes. The nematodes that live in the Antarctic and other cold places also have really interesting biology. And this is an example of that. So these nematodes have evolved a special adaptation called anhydrobiosis, where they're able to push all the liquid water out of their body and essentially become freeze-dried. And when they become freeze-dried, because they live in places like Siberia or the Antarctic dry valleys where liquid water is not available most of the time, they can suspend their metabolism by freeze-drying themselves. And scientists believe that they may be able to survive in those conditions indefinitely. And here's a report of some scientists who found some dried-up nematodes in permafrost that they dated to over 40,000 years old. And when they added liquid water to those nematodes, they rehydrated and they essentially came back to life. Sounds like something out of a science fiction movie, like the beginning of a horror film, but that's real. That's real life. That's biology. This is a famous entomologist who I love, Edward O. Wilson, and he said this: consider the role of nematode worms in maintaining the health and fertility of soils. We don't know how many nematode species there are. We do know they must be important. We don't know what they're doing. Consider this: 4 out of every 5 animals on Earth are nematode worms. That's shocking, and it's true. Everything he said is true. If you were to take all the animals on planet Earth and put them into a large bag and pull 5 out, just due to their abundance, their sheer abundance, 4 of them would be nematodes. So with that introduction to general nematodes, let's take a minute and let's talk about entomopathogenic nematodes. These are some nematodes that are dear to my heart. They were the first nematodes I actually came to learn about, and I started studying as an undergraduate. These are parasites of insects, and they're very successful parasites of insects. So what makes them entomopathogenic, which is a subset of insect parasitic nematodes, is that they kill their hosts very quickly, usually within a few days, and they utilize bacteria to facilitate their parasitic lifestyle. So if you imagine this is a caterpillar living in the soil, these nematodes can enter this caterpillar through natural openings, through the mouth, through the anus, and once they get inside that caterpillar, they will spit out venom proteins, and they will release highly pathogenic bacteria into the blood of this insect. And so within a day or two, the insect dies, and these nematodes develop to adulthood, they reproduce, and when all the resources of the insect are eaten, you have tens of thousands of parasitic nematodes that will emerge out of that insect cadaver. And all these nematodes now are in the soil looking for more insects that they can infect and kill. This is really a great system. When I first learned about this, I thought this could be like something out of a James Cameron movie. You've got this worm, it crawls inside of its host, it spits out toxic venom, and then to add insult to injury, in the case of the nematodes I study, they actually defecate out highly pathogenic bacteria into the blood of that insect, and together the bacteria and the venom kill this insect, and they liquefy the insect tissue and convert that insect down to nematode biomass. So what starts as maybe 10 to 20 nematodes that initiate this infection, 7 days later, 10 days later, you may have 60 to 80,000 nematodes that come out of that. And all of them are going to be in pursuit of new insect hosts to infect. So at the top of this life cycle, I'm showing the stage that lives in the soil, and these are going to migrate into the insect. That's the infective juvenile life stage. It's developmentally arrested, and its mouth is actually sealed. So it's in the soil looking for an insect to infect, and when it gets inside, it has to become activated. It initiates the active parasitic stage of its life cycle, and it starts to resume development. And when it makes that transition is when it spits out venom and when it starts to worry about dealing with the immune response of its host, okay? And that behavior is not unique to entomopathogenic nematodes. That's true for some parasites of humans, like hookworm. They live in the soil as an infective juvenile. Their mouth is sealed, and when they penetrate through the skin of a human, they activate the parasitic stage of their life cycle, and they spit out proteins and other things, and they begin to interact with the immune response of the host. So here we're looking at a video of an insect that has died, been dead for 5 to 10 days, and what you're seeing, it's not top ramen, it's nematode worms that are just gushing out of these insect cadavers, and they're all put together in close contact like muscle fibers. And these guys are going to be migrating their way into the soil looking for new hosts to infect. And if you're thinking to yourself, that might be useful in agriculture, you're right. We use these nematodes as an organic pesticide or as a form of biological control. These are sprayed by the trillions in orange groves, soy fields, corn, all different kinds of crops, and not just in the United States. These are used globally. In fact, you could buy them at the local hardware store or online as beneficial nematodes that could be sprayed into your home garden as a way to prevent insects from damaging your garden. So in the soil, you have these nematodes that are living there looking for insects to infect. And in the genus Steinernema, which are the nematodes I spend a lot of time studying, there's about 100 described species, a little more than that. Some of them are going to migrate into the soil, and they're going to be really good at finding insects that live in the soil. Some of them are going to be living at the soil-air interface, or the so-called epigeal habitat, where they're interested in attacking highly mobile insects like crickets or earwigs or things like this, and they adopt kind of an ambush foraging strategy. So they're standing on their tails, and they can initiate some really interesting behavior. One of the things that they're capable of doing is they can actually jump and attach onto a passing insect host. I want to just say, only insect parasitic nematodes are known to be capable of jumping, and only certain ones. No parasites of humans have been found to be capable of doing that. But it's such a striking and interesting behavior. I want to show you a couple of videos. This is one I took as a graduate student, and you're looking here at a nematode standing on its tail. This is a very small worm. It's less than a millimeter in length. It's about 600 to 700 microns in length. And when it gets touched by that insect, you can see it coils down and it jumps. It's hard to make that out in this video because this is filmed in real time. I've been interested in this behavior since I was a graduate student. And just this year, in collaboration with a few colleagues at Georgia Tech and UC Berkeley, we have studied very carefully the biophysics and biomechanics of jumping behavior, and we have identified the forces involved, how nematodes that have no hard body parts or legs are capable of doing this kind of jump. And my colleagues have really expensive camera equipment, and I want to show you some of the best videos that they've taken of this behavior because I think it's so interesting. So this is a nematode that's coiled up and just ready for the moment of liftoff. And this is filmed at a rate over 1,000 frames per second. Sorry, over 100,000 frames per second. Yeah. So this is filmed at a really high rate of speed, and you can see just the moment that this thing takes off and launches itself, it can jump 20 times its body length. I mean, this thing can really, if you could jump that high, you would be an Olympic athlete for sure. I have a video here where you can see, if you look at the bottom right on this little piece of paper, you can't see the worm yet, but it's going to take off. And it's going to somersault like crazy. And again, this is filmed at over 100,000 frames per second. So it looks like the film stops here, but it's not. It's just gravity takes so long that it almost looks like this is just hanging out in the air there until it starts to come back down. No. I want to go back so you can see that one more time, because I just think that's so cool, and that my collaborators were able to capture this behavior at such a high frame rate. What an incredible thing that these nematodes are capable of doing. The fact that they can just jump 20 times their body length. Okay. For the remaining of my talk, I want to spend just a few minutes talking about how parasitic nematodes cause disease and some of the research we're doing here. So in these pictures, I'm showing you the same species of nematode. On the left, you're looking at what the infective juvenile looks like when it's in the soil. So this is done by scanning electron microscopy, and these images were actually taken here at UCR. So in the soil, the mouth on this nematode is sealed. You can see it's sort of puckered there, and it's non-feeding. It's also halted its development, and in that form, it's looking for a host to infect. And on the right, we've taken this nematode and we put it in insect tissue for about 12 hours, and in that time, you can see the dramatic morphological change that takes place as it initiates the active parasitic stage of its life cycle. So now its mouth is open wide, it's spitting out toxic venom into the host, it's defecating bacteria into the host, and it's resuming development. It's going to develop to adulthood, and it's going to reproduce. And this is the same change that hookworm undertakes in its human host, that threadworm undertakes in its human host. And so by studying this process in these nematodes, we're hoping to learn something about the initiation of parasitism by nematodes in general. Now, this is a very tricky thing to study. If I told you I wanted to study snake venom or spider venom, I could probably get a significant amount of protein from an individual, from one spider or from one snake. If I told you I wanted to study the venom of a nematode that's less than a millimeter long and like 20 microns wide, how many nematodes do you think I need to milk to get that venom? It turns out it's a lot, and it's a lot of work. So I want to recognize some of my students who've done this work. And so we're essentially trying to trick these nematodes in a petri dish or in vitro to start secreting this toxin, this venom, so that we can collect it and we can do subsequent studies with it. And so the way we do that is we take waxworms or other insects and we make a soup out of it. I tell the students in the lab all the time, this is not broccoli cheddar. It looks like that, but this is waxworm homogenate, and the nematodes love it. So we put the nematodes in this insect soup, and they activate and they start to spit out their venom. And you can see here some of this blue dye that we've used to show this nematode on the far right spitting some of its toxic venom out into clean buffer. So the way that we do that is we put the nematodes into this insect homogenate for some amount of time. Once they start spitting out this venom, we can pull them back out of that. We can wash them off very carefully and put them into a clean buffer where they continue to spit out this venom, and then we can collect it and we can start to do studies with it. And I can tell you to get 8 milligrams of protein, okay, which if you guys, anybody here takes a scoop of protein powder in the morning, that's like 30 grams, right? I'm talking 8 milligrams, right? And we end up getting, it takes 100 million nematodes to get that much protein. That's a lot. That's a lot of work. So what we've been doing with that is we collect this venom and we decide we want to see what it does to an insect. So we can inject it into tiny little fruit flies, and that means we don't have to inject that much protein. This is a difficult skill to learn, I can promise you. Some of the students in my lab have injected themselves more than 30,000 fruit flies. Imagine how long that takes. That takes a long time, and they're very tiny. We've learned that one individual nematode in the course of a 24-hour period may be able to secrete about 0.5 nanograms. So if you had 100 nematodes in one of these infections, you may get 50 nanograms. A nanogram is like a millionth of a milligram. So we're talking a little tiny little amount of venom, and we inject this into these insects. So I'm going to walk you through. I've got these 2 data graphs here. This is a Kaplan-Meier curve. It's a survival curve. We're asking, do the insects die when we inject them with this venom? So on the y-axis is the percentage of the flies that are alive. And so it starts at 100% when we start the experiment. And the x-axis is showing you time, either hours or days after we've performed this injection. And what you can see, so the different colors, we've exposed the nematodes. This is how long they were exposed to insect tissue for. 6 hours, 12 hours, 18 hours. What we're showing here is that when you inject a fruit fly with 20 nanograms of this venom, within a couple hours, they're dead. They die very fast. And when we use venom or protein secretions from a nematode that has been in the homogenate for 42 hours, it's close to adulthood by that time, and the types of proteins it's spitting out change, and they're no longer toxic to these insects. So we discovered that these nematodes their venom is insecticidal. It's highly toxic to insects. Now here, we've injected these caterpillars with plain buffer, and we looked at it 24 hours later to show we can inject them, and just the act of injecting them doesn't hurt them. But if we inject them with a little bit of this nematode venom, and we look at how well they're doing 24 hours later, I can show you they're not doing well. We begin to see necrotic tissue. They're turning black. They're not very responsive. These are not happy campers. And so we've shown that this nematode venom is actually toxic to a wide variety of insects, that it can kill them. So we had this question then. We know that entomopathogenic nematodes are safe for humans. So you can buy them at Home Depot. You don't need any kind of special permit. We know they don't cause any disease, but we wanted to know, does the venom that they secrete, can it affect mammalian immunity? And so we incubated mouse macrophages with some of this venom, and then we introduce a bacterium that is stained with a fluorescent dye that will only fluoresce red if that bacterium gets engulfed by a macrophage. So we can measure phagocytosis by macrophages, and we want to see, does this nematode venom influence mouse macrophage activity? And I have some data to show you, just a couple pieces. So here in blue, we've tracked the nucleus of the macrophages. So all the dots that you see that are blue, those are the cells. And in the bottom left hand, you can see some red. That's bacteria being phagocytosed. That's bacteria being taken in by macrophages. These mouse cells were just exposed to clean buffer. When we expose those same macrophages to this venom, and we do the same experiment, we get a lot more red. It's like the macrophages are being primed. It's like they're being activated so that they're more ready to gobble up bacteria. And here we can quantify that. And that's really interesting because it means that this nematode venom, even though these are parasites of insects, the components of their venom have an effect on mammalian immunology. I'm getting to the end here. I want to come back to what's in this venom. So we know it's a complex mixture of more than 500 proteins, or around 500 proteins. Now we can separate those proteins using a variety of biochemical techniques. We can fractionate it. We can identify the components, and we can learn about what the individual pieces do. And one of the cool things is one of the proteins we found here, we think it has insecticidal capabilities. Just one of the 500 proteins that are in this venom. So what can we do with that? Well, we can feed it to insects and ask, is this one protein capable of killing insects? We can also make a plant that can express this protein, which we've done. We've made tomato plants that express this nematode protein, and then we've allowed the fall armyworm, this voracious plant feeder that destroys crops, we've allowed it access to these tomato plants, and we can ask, do the insects like to eat this plant, and do they do well when we allow them this opportunity? And it turns out they don't do well, and they don't feed on these plants. So this is really interesting. This venom that we've collected from these nematodes may have biological application and commercial application that could benefit humans by protecting our crops from insects damaging them. I mentioned at the beginning of my talk that nematodes cause considerable human suffering. I'm showing here a list of some of the most prolific parasitic diseases, how many people are infected, how many deaths are caused each year. And in this red box, I'm showing you the nematodes. Okay, so Ascaris lumbricoides, that's one species of nematode. It infects over 1 billion people, and that's true for today as well. Hookworms, that's a complex of multiple species, as are filarial worms. But you can see we're talking about billions of human beings infected with parasitic worms. There's a lot of questions there. How do they cause disease? How can we stop them? Do they modulate host immunity? One of the things that really I think is most incredible about these nematode infections is that most of them are asymptomatic. You can see the number of deaths that we're talking about compared to things like malaria or flukes, and the numbers are much lower. I want to share one case study with you that I share in my parasitology class that I teach. It's about this 21-year-old woman. She's living in England, and she goes to see her doctor because she thinks that maybe she can feel something moving in her eyeball. And so when she meets with her doctors, of course, they ask lots of other questions. Does she have any pain? Is she tired? Does she have any other symptoms or pathology? And it turns out that she doesn't. She feels otherwise perfectly healthy. Her medical history is only noteworthy for the fact that she took a trip 6 years earlier to Nigeria. And when they examine her, indeed, they do find a 2-centimeter long worm in her eyeball. They're able to surgically remove that. But when they take a blood sample from her, they find that she has tens of thousands of parasitic worms in her blood, in her musculature. I mean, she is heavily infected. And what's shocking to me about this case is she has no pathology. She's not sick. She's not complaining about pain or any other pathologies. The only reason she went to see her doctor is that she felt something moving in her eye. I'm going to show you a picture of that, but I'm warning you in advance in case you don't want to see it. I won't leave it up on the screen for very long, but I think it's worth seeing. So this is if you were in my parasitology class, we'd watch the video of them surgically dissecting this. I won't subject you to that, but it's a good video. So my point is these nematodes are capable of modulating host immunity to the point that most of the infections are asymptomatic and the immune system doesn't even know that it's happening. I had just a couple more pieces of data to share, but I can see that we're getting to the end of it. What I just want to conclude by saying is that nematodes have a significant impact on humans and on our environment. Entomopathogenic nematodes, which I've been studying and I've shared with you today, have a beneficial effect of protecting our crops. They can kill insect pests and they can keep our crops safe, and they still have a lot to teach us about how to manage insect pests. And lastly, parasitic nematodes are present in force on this planet. Many human beings are infected. They do cause significant human suffering, but they also have a lot to teach us about how the immune system works and how the immune system can be manipulated. I want to thank you for your time today. I want to thank you for coming to my talk. I want to thank the people who have done some of this work in my lab, and of course the funding agencies, which Peter has mentioned at the beginning, and I would be happy to take any questions that you might have. Thank you.
Thank you, Adler. If you have any questions, please raise your hand. Now, for that question, the microphone.
Hello.
Hi.
I was wondering if there is any major ecological impacts of like mass release of nematodes into the environment, especially ones that are mass cultivated by corporations.
That's a very good question. As I mentioned, these are dumped into the environment by the trillions in certain areas in agricultural crops. The dogma in the field is that there are little to no off-target effects, and the environmental or ecological impact is minimal. I would say, as someone who's read a lot of that literature, I think more work needs to be done. They do seem to have a significant impact on certain insects first. Like if you look at when you release these into the environment, which insects go away first, earwigs are one of the insects that just kind of disappear. And so I think that there may be some ecological effects, but right now they're not very well studied and not very well known.
Thank you.
Good question. They'll bring a microphone over to you. Okay.
What happens when they run out at the end of it? That's feedback.
So as I mentioned, in the soil, they're infecting juveniles with a sealed mouth. They have a certain caloric reserve, and if they can't find a host to infect within a month or two, depending on environmental conditions, yes, they will die. That's it. Yep. Yes.
Question about the nematode jump. Is there a trajectory or is it planned? Is it just a response to stimuli?
It's a very good question. They can jump forward and they can also jump backward. And it seems like they do have some directional control of that behavior. It's kind of hard to study it in the lab because it's artificial, right? It's not the soil, especially if you want to see them. You can't do this experiment in soil and still see it. So we're doing it on filter paper and other clever media that we can make. And it looks like they do. So if you take a puff of carbon dioxide, if you puff CO2 from a direction, they can jump, and it seems like they can maybe jump even in that direction. In addition to looking for hosts, it's speculated that this may also serve as a means of dispersal. So they're very small and they can't move very fast or very far, but you can imagine if they could jump and catch a breeze or something, you might have a lot further distance that could be traversed by jumping than by migrating through the soil. Great question. Thank you.
Good.
All right.
So you mentioned that like a lot of nematodes are a big threat to insects. Do you know if any insects have developed any kind of like defense mechanism to the nematodes?
That's a great question. So certainly insects have a sophisticated immune response. And a lot of the insects we study in the lab are soft or weak insects. So ones you can buy at the pets shop that you might feed to your reptile, those will die very easily to these nematodes. I've studied other kind of hardier insects. We've studied some mole crickets. There happens to be a population of mole crickets in Downey. They're not supposed to be there. They've come from ornamental plants. They're very hard to kill. And so if we try to infect them with some of these nematodes, they can kill them at a very low rate. So there are some insects that seem to be very hardy and resistant to their infection. In terms of the strategies to get rid of them, I mean, so they have sophisticated immune responses, but some of them have adopted certain grooming behaviors. I noticed, again, just going to the mole crickets, some of the mole crickets have a blade-like structure on one of their forearms, and they can scrape their body with this blade as a way to sort of groom, and it will actually kill those worms that are on them. So there may be some behavioral adaptations too. Yeah. Very good question. In the middle.
So I know that you said that there's a lot of different types of the nematodes. Has the discovery of new species or new types of it stagnated with the recent years, or has it been kind of new ones every year? So
that's a good question. So Peter can tell you there's a lot of people that study insects. There are a tiny fraction of that studying nematodes. Far fewer people are studying nematodes, and there are still new species being described. But some of the skill that's necessary to describe the physical features of the nematode under the microscope, to do the measurements and to measure the certain body parts that we do in addition to the molecular work, that's a skill set that is waning. So there are fewer people that have that particular skill set, and it's being taught less and less. That being said, since I've been at UCR, we've described a couple new species just in the last 3 years. So we are actively still looking. And when people call us with something that they think is interesting, we're usually quick to respond. We've described a new parasite of tarantulas, a new species of nematode that infects tarantula spiders. And we've also described a new species of entomopathogenic nematodes that someone shared with us. So there are still new species being described, but I would say it's probably less than at earlier times in the study of nematodes.
You mentioned earlier that some of your nematodes of interest had bacteria internally that they would defecate into their host. How did they obtain that bacteria?
That is a really good question, and a lot of people so there are more people studying that bacteria than there are studying these nematodes, and that's it's open for discovery. So what I can tell you is that the bacteria they house, they've evolved a special structure in their throat to house it. You don't find that bacteria anywhere else on the planet. It's never found in the soil. It's not in the water. It's never been found outside of an infected insect or in these nematodes themselves. So where it comes from is a very good question. We know some of it is closely related to some of the bacteria that's in deep water cephalopods, Vibrio fischeri. There's a close relation there with some of the bacteria, but where it comes from is a totally open question and one that you could potentially study. Thank you again for all of your questions. I appreciate it.
So I'll just make 2 comments. One, first is a very local and sort of semi-irrelevant one, but I run a laboratory with a colleague in botany and plant sciences, and as part of that project, we have to grow lots of living brassica plants. We order lots of things in our laboratory, enzymes, test tubes, etc. Many things we order. There's only one thing that we have a standing order for, and that is nematodes. So every 2 weeks, we get in a supply of nematodes from a company in Thousand Oaks, somewhere like that. And that enables us to keep our plants alive. Now, that is a fairly peculiar local example without any meaning beyond our laboratory. but in fact, it's the only standing order we have. The bigger implication is, as Professor Dillman said, is that we live on an amazing planet. I mean, the things that he described, most of which we can't see with our naked eye and we don't know about, and that extends to billions of organisms if we count them, and millions of species. But on this amazing planet, which we are guardians of, as perhaps the most dominant species, it's our job to make sure that that ecology, that entire ecosystem continues. The planet also has amazing professors and faculty, so we've been blessed to have a seminar by one of those. So thanks again, Dr. Dillman. This concludes our Science Lecture Series for 24-25. We'll be back in business in 25-26, so please keep an eye on the CNAS communications page for the titles of those seminars, the professors who will present them, and there'll be at least 2 down here, maybe more, and we hope to see your participation next year in our series. So thanks again for coming out today. Have a safe trip home, and look forward to seeing students next year, and looking forward to seeing others, parents, faculty, students, anyone coming to our Science Lecture Series for next year. Thank you so much.